DNA structure and replication. In 1953, double helix model of DNA is proposed based on the primary information, that is the X-ray diffraction studies of Rosalind Franklin and Wilkins. So the DNA having double helical structure consisting of 4 bases, adenine and thymine, guanine, cytosine. Each base pair, a gap is there in between 2 base pairs in the form of the 0.34 nanometers. And we can measure the length of the DNA by taking each turn in the form of the 10 base pairs, 3.4 nanometers or 34 angstroms. So the DNA exists in 3 different forms: A form, B form, Z form. The B form is the most common form and right-handed double helix, and Z form is the left-handed double helix. The right-handed, which is containing the 10 base pairs, left hand which is containing the 12 base pairs. The dehydrated form is the A form. So the right-handed differs from the left-handed by glycoside bond variation in the form of the anti-form in the whatever the glycoside bond in between base and sugar in case of the B form, A form DNA. And Z form DNA, the glycoside bond is in the form of the syn conformation, that is getting the zigzag form of the DNA. So the structure determination of the DNA is given Nobel Prize in 1962 for Watson and Crick and Wilkins. The structure of the DNA, which is containing the 2 strands in the form of the opposite polarity and consisting of 4 bases in the form of adenine, thymine, guanine and cytosine. Always one purine pairs with the pyrimidine and pyrimidine pairs with the purine, vice versa. That is the Chargaff's rule. So basically we can see that there are 2 complementary strands in the form of the 5-2-3 and opposite polarity, and they will pair with the 2 opposite polarity strands. So the DNA replication is proposed, hypothesized in the form of the 3 methods, that is the semi-conservative, conservative, dispersive. In case of the semi-conservative, one strand is old, the other one is the new one. In case of the conservative, both the strands are old or new one based on the new one replicates based on the old one. In case of the dispersive, we can see here and there in the form of the old and new strands. The Michelson and Stahl, their classical experiment in 1958, they proven by culturing the E. coli in the N15 medium of nitrogen isotope and N14. They cultured in pure N15 and transported into N14 or N14 to N15. We can see the in between, we can see the an hybrid strand in the form of the N15 and N14. So whenever we are doing extracting the DNA from the cesium chloride gradient and we do the centrifugation at a high RPM at 1 lakh RPM at 50 to 60 hours, we can see the either pure form N15 and N14, and we are transferring in between N15 to N14. We can see the hybrid strand in the form of the N15 and N14 strands. That indicates that the DNA synthesis is taking form in the semi-conservative. So we can see the DNA replication takes place. Origin for the replication, we can see the a bidirectional replication in the form of the theta-like structure. We can see the 2 opposite directions in the, we can see the one old strand, new strand in the form of the yellow and pink colors. The replication is semi-conservative and requires a template. So the DNA precursors in the form of the DNTPs, deoxynucleoside triphosphates, in the form of the ATP, TTP, CTP, GTP, like that, for the addition of nucleotides to and synthesizes the nascent strand. Replication in E. coli, the nucleotides added only the 3 end of the growing nascent strand. Therefore, the nascent chain grows only in the 5-3 direction. So additional nucleotides are added the elongation direction. So replication in E. coli will be takes place by the three enzymes in the form of the DNA polymerases 1, 2, 3. So in 1950s, Arthur Korenberg discovered the first DNA polymerase 1 enzyme that is having the three sites in the form of the 5-2-3 elongation and 3-5 exonuclease activity for proofreading and 5-3 exonuclease activity for the excision of the RNA primers. So the DNA polymerase 1 lacking 5-3 exonuclease is known as the Klinos fragment. Korenberg's. Based on the replication, the scientists thought that the DNA having polymerase having the three enzymes rather than the only one enzymes. In 1970s, DNA polymerase 2 and 3 were discovered. 2 and 3 is lacking 5-3 exonuclease activity and having 5-3 polymerase activity for elongation, 3 exonuclease activity for the proofreading. DNA polymerase can only elongate the existing chains. It cannot synthesize the de novo chain synthesis. Nascent strand initiation requires formation of the short RNA primers. RNA primers synthesized by the one of the primase enzyme in the 5-3 RNA polymerase, initiating the nascent chain from the DNA templates, uses the ribose NTPs as nucleotide source. The primers are eventually excited by the 5-3 exonuclease activity of the DNA polymerase 1. Replication in E. coli. The replication begins location chromosomes called origin for the replication. The DNA helix unwinds the origin, 2 old strands become 2 distinctive templates in the form of 3-5 template and 5-3 template. So the replication on the 3-5 templates continuous synthesis of the DNA taking place, that is known as the leading strand. On 5-3 template, discontinuous synthesis of the short nascent segments, lagging strands or Okazaki fragments, each with its own primer. After primer excision, complete nascent segments are sealed by using one of the DNA ligase enzyme. The DNA polymerase may have able to synthesize both the leading and lagging strands simultaneously by having the 5-3 template fold back. So we can see there are 2 core enzymes in the DNA polymerase 3, and each core enzyme containing the 3 subunits in the form of the one of the alpha subunit, which is synthesizing 5-3 polymerase activity, one of the epsilon, which is useful to proofreading activity in the form of the 3-5 exonuclease activity, and theta, which is useful to activate the exonuclease activity. So by using these 2 core enzymes, the one of the strand which is synthesized in continuous fashion, that is the leading strand, and the other strand which is synthesized in the 5-3 direction in the form of the discontinuous fashion, that is known as the Okazaki fragments. So always the DNA synthesis will needs a primer which is synthesized by the primase enzyme, so which uses the NTPs in the form of the RNA will be synthesized by using a DNA primase enzyme. Then it will be extended by the 5-3 polymerase activity of the DNA polymerase 3. So whatever the RNA primers are there in the lagging strand, they will be either digested with the RNase enzyme or removed by the DNA polymerase 1 5-3 exonuclease activity. The RNA primers are removed and the DNA synthesis will be takes place by the 5-3 polymerase activity and 3-5 exonuclease activity, which is useful for the proofreading. So whatever the breaks are there in between these 2 fragments sealed with the a ligase enzyme, and that is forms the phosphodiester bond. So we know that one of the DNA B gene which is useful to separate the two DNA strands, that is known as the DNA helicase enzyme. So it is synthesized by one of the DNA B gene. With the help of the ATP, it is separating the two strands. And we can see the different types of the enzymes in the form of the primerases and helicases and single-stranded binding proteins in the form of the topoisomerases. So the primerases which are synthesizing in the form of the RNA primers, helicase which is MCM complex or DNA B gene helicases which separate the two DNA strands, and we can see the single-stranded binding proteins in the form of the replication protein A or single-stranded binding protein. And we can see the whenever the DNA synthesis is taking place on one direction, another direction the supercoils will be formed. These supercoils will be removed by the nicking of the one of the DNA strand with the help of the topoisomerases that will be unwinded. So the DNA guidages makes temporary single-stranded nicks in one of the two template strands, relieve the torsional stress and supercoiling caused by the unwinding of the helix. And we can see the single-stranded binding proteins bind to unwound strands template, stabilizing the single-stranded state long enough. And we can see the different enzymes in the form of the topoisomerases, two guidases, which are useful to break the nick the one of the strand, and it removes the supercoils. And DNA primase, which is useful to synthesize the RNA primers, and helicase DNA B, which is useful to separate the two strands, and whatever the leading strand which is synthesized by DNA polymerase 3, and also we can see the RNA primers and the synthesis of the lagging strand in the 5 to 3 direction, and it is binded with the single-stranded binding proteins. And we can see so after the synthesis of the two DNA strands will be separated in case of the prokaryotes by using the topoisomerase 2. It breaks the two DNA strands. So like this we can see in prokaryotes one of the DNA polymerase 3, which is the important DNA polymerase enzyme. It is having the two core enzymes which is having the three subunits. One of the subunit, alpha subunit, which is synthesizing the DNA in the 5 to 3 direction, that is synthesizing the nascent strand, and epsilon, which is useful for the proofreading to remove any mispairing bases, and theta it will be activates. So like this we can see in addition to that we can see the DNA polymerase 1, which removes the RNA primers, and it fills the 5 to 3 polymerase activity in the gaps, and after that whatever the strands which is having breaks will be sealed together by using the ligase enzyme. So like this we can see the DNA duplication which is taking place in the semi-conservative way. What Michelson and Stahl proposed in 1958 by using the N15 and N14 media, and we can see the three important DNA polymerasing enzymes. Out of these, three is important, and it is having three sites. It is synthesizing both the strands at a time, and in the form of the leading strand and the form of the lagging strand in the discontinuous fashion. The RNA primers which are removed by the DNA polymerase 1, and it is filled with the 5 to 3 polymerase activity, and after that they will be stick together in the form of the ligase enzyme. So this is the DNA replication enzyme we can conclude now.